So hello, and thank you very much for visiting my talk. My name is Kerri Kinghorn, and I'm based at the Institute of Healthy Aging at University College London. And today I'm going to be speaking about the gut and its microbiome and the role that it plays in triggering an innate immune response in a Drosophila model of GBA knockout. So first of all, what is the GBA1 gene? So the GBA1 gene encodes a lysosomal enzyme known as glucocerebrosidase, or GCase for short, and this is responsible for the hydrolysis of its main substrate, glucosylceramide, to ceramide and glucose. And it's been known for a number of decades now that homozygous or biallelic GBA1 mutations cause the commonest lysosomal storage disorder called Gaucher disease. So this is a multisystemic disorder which is characterized by the accumulation of glucosylceramide within macrophages, which then become deposited within various organs such as the liver, the spleen, and the bone marrow to cause organ dysfunction. And rarely there is also brain involvement in neuronopathic forms of Gaucher disease. Now, one of the reasons I became particularly interested in this gene was because in recent years it has become established that heterozygous mutations in GBA1 are the commonest genetic risk factor for developing Parkinson's disease, conferring up to an approximately 20-fold increased risk. So in order to study how GBA1 loss of function causes neurodegeneration, I previously developed a fruit fly model of GBA knockout. Flies have two GBA1 genes. They have GBA1A, which is expressed exclusively in the fly gut, and also GBA1B, which shows widespread expression, including in the gut, in the brain, and also in the fat body. The fat body being the equivalent of the mammalian liver and adipose tissue. So given the fact that GBA1B is the main fly ortholog of GBA1, I previously knocked out this gene using homologous recombination, and it's this model that I'm going to focus on in this talk. So one of the first things I then did was to ensure that this was a faithful model of a lysosomal storage disorder. And so using mass spectrometry on the heads of GBA knockout flies and controls, I showed that there was accumulation of the GK substrate glucosylceramide within the heads of the GBA mutants. And then using LISA tracker and staining of the brains, I showed that there was an increase in the number and the size of the lysosomes in the GBA knockout fly brain compared to controls, consistent obviously with the fact that they're engorged and swollen up with glucosylceramide. We performed lots of different characterization on this fly model. We showed that flies lacking GBA1B are very sick, they have reduced lifespan, and we also showed that they displayed age-dependent locomotor decline. Downstream of the lysosomal defects, we showed that there was a significant block in autophagy in the fly brain, and that this was associated with accumulation of proteins such as P62 and polyubiquitin. The mitochondria also appeared very abnormal, and there were reduced levels of ATP. And all of these defects led to neurodegeneration and synaptic loss. So in order to further study these flies, we then performed bulk RNA sequencing on the heads of GBA knockout and control flies, and we did this at 1 and 3 weeks of age. And gene ontology analysis revealed that there was gene enrichment and upregulation of genes that are linked to molecular pathways and biological processes mapping to innate immune signaling, particularly at 3 weeks of age. And so then we turned our attention to the immune system of GBA knockout flies. So many immune pathways are conserved between mammals and flies. And just as humans have humoral innate immune responses, so do flies. So flies have two NF-kappa beta pathways, the toll and the IMD, or immune deficiency pathway, which correspond to the toll-like and the TNF pathway in humans. And then there's also a highly conserved JAK-STAT pathway. And all three of these pathways respond to stimuli such as fungi, bacteria, and viruses, just like in mammals. And here in red, I have highlighted those pathway mediators that were upregulated in our RNA sequencing in GBA knockout fly heads. Now, downstream of all of these pathways, you get upregulation of a number of response genes. So downstream of the NF-kappa beta pathways, the toll and the IMD pathway, you get upregulation of a number of antimicrobial peptides. So, for example, one of those is drosomycin downstream of the toll pathway, and diptericin, which is downstream of the IMD pathway. And then downstream of the JAK-STAT pathway, a number of stress response genes are upregulated, such as TOT-A, as shown here. And when we looked at gene expression levels of all three of these genes in the heads of GBA knockout flies, we saw that all were increased in the mutants compared to controls, consistent with all three of these innate immune pathways being increased in GBA knockout fly heads compared to controls. We then turned our attention to a different form of innate immunity, that being cellular immunity, and we focused particularly on the function of a well-conserved glial immune pathway, which involves a receptor called DRAPER, which is MEGF10 in mammals and humans. So DRAPER is a phagocytic receptor which is found on the surface of glia in the fly brain, and it's responsible for the phagocytosis of cellular debris. And therefore, it's considered a good marker of glial activation in the fly brain. So consistent with humoral innate immune pathways being upregulated in the heads of GBA knockout flies, we also saw that there was an increase in DRAPER at the transcript level, at the protein level on Western blot analysis, and also on immunostaining with an antibody against DRAPER in the GBA knockout fly brain, consistent overall with there being an increase in cellular immunity in GBA knockout flies and increased glial activation. We also looked at the humoral innate immune pathways in many different tissues in GBA knockout flies, and we showed that there was an increase in innate signaling in all of the tissues that we studied, including, as shown here, in the gut, where you can see there's an increase in toll pathway activation and also IMD, innate immune pathway activation. And this was particularly interesting to us because the gut is increasingly being shown to play an important role in a number of diseases, including neurodegenerative diseases such as Parkinson's disease. And consistent with gastrointestinal symptoms being an almost universal feature in Parkinson's disease patients, intestinal inflammation and increased gut wall permeability have been shown to occur in Parkinson's disease. Moreover, numerous studies have shown that there are alterations in the gut microbiome in Parkinson's disease patients compared to healthy controls. The communication between the gut and the brain, the so-called gut-brain axis, is likely complex and mediated by direct neuronal communications, but also by systemic mediators such as immune, hormonal, and metabolic mediators. The question, obviously, that we were interested in is, does the gut and its microbiome play a role in GBA1 associated neurodegeneration? And so to study this, we turned our attention to the gut of GBA knockout flies. So one of the first things we did was to look at intestinal transit. So we counted the number of fecal deposits produced by the flies after a meal. And what we found was that there was a decrease in fecal deposits in GBA knockout flies within a few hours of eating a meal, but that in the ensuing hours, the deposits were then increased in the GBA knockout flies compared to controls, consistent overall with an increase in intestinal transit time and a constipation-like phenotype, as you see in patients with Parkinson's disease. We then turned our attention to intestinal wall integrity and barrier function. And to do this, we use something called a Smurf assay. So flies are fed with a non-absorbable blue dye. And if that dye is able to pass across the intestinal wall into the surrounding tissues to give a blue Smurf-like appearance, then that indicates that there is increased gut wall permeability. And indeed, we saw an increased number in Smurfs in the GBA knockout fly group compared to controls, consistent with there being increased gut wall leakiness in GBA loss of function. We also looked at the gut microbiome, and we used two methods to look at overall gut bacterial load. One was essentially quantifying bacterial DNA, and the other was counting colony-forming units of bacteria. And both of these methods showed that there was increased gut bacterial load in GBA knockout flies compared to controls. We also looked in more detail at the microbiome using 16S ribosomal RNA sequencing, and this showed that there was significant alterations in a number of gut species between the controls and the GBA knockout flies, consistent with there being an overall alteration in the gut microbiome in GBA knockout. The next obvious question we wanted to ask is what would happen if we completely eradicated that gut microbiome? And so in this case, that means rearing the flies under germ-free conditions and treating them with a cocktail of antibiotics. And to our surprise, what we found was that GBA knockout flies that were reared under germ-free conditions had an innate immune response in the gut wall that went back down towards control levels. And this was also associated with an increase in survival. So the GBA knockout flies reared under germ-free conditions had a better survival than those reared under standard conditions. So then what we wanted to do was to establish what was underlying the gut wall abnormalities. Now we know that there are defects in the lysosome and in autophagy in GBA knockout fly brains, and we wondered whether the same might be happening in the fly gut as well. And so we looked at autophagy. Autophagy is the process by which defective organelles or misfolded proteins become enveloped in a double membrane within a structure called an autophagosome. And then that fuses with a lysosome to form an autophagolysosome. And it's within this structure that the contents are then degraded by lysosomal hydrolases. And we can monitor autophagy by looking at a number of proteins that are associated with this process, including Ref2P, which is the equivalent of P62 in mammals, and which is an adapter of autophagy, and also ATG8A, which is the same as LC3 in mammals, and which is a marker of autophagosomes. And by looking at the levels of both of these proteins, we were able to show that both were increased in the gut wall of GBA knockout flies, suggestive of there being a block in autophagy. And we saw this also under germ-free conditions and normal conditions. So then what we wanted to do was to see, well, what would happen if we tried to reverse the autophagy defects by feeding flies with the mTOR inhibitor rapamycin, which stimulates autophagy. And indeed, GBA knockout flies that were treated chronically with rapamycin showed a significant prolongation in their lifespan compared to those that were untreated. And interestingly, we also saw that this degree of lifespan extension was very similar to that achieved under germ-free conditions. Moreover, what was most surprising was that if we raised GBA knockout flies under germ-free conditions and also treated them with rapamycin simultaneously, that this had no additive benefit on lifespan, consistent with both interventions acting in the same pathway. And we saw a similar effect also in terms of these treatments on the innate immune response in the gut wall of GBA knockout flies, with all treatments reducing, but no additive benefits from the dual interventions. And so what do we think is causing this chronic innate immune activation in the GBA knockout fly gut? Well, we know that certain components of these innate immune pathways are kept under basal levels by removal by autophagy. And so if there is a block in autophagy in the tissues of GBA knockout flies, particularly here in the gut, then these innate immune mediators increase, and this leads to constitutive activation of the innate immune responses. So this is what we think is happening in GBA knockout flies. And so just to summarize what I've been showing you today, we have shown that there is gastrointestinal dysfunction in flies lacking GBA1, and that this is associated with a leaky gut wall and an altered microbiome. There is increased inflammation and increased immune activation in the gut tissue. And because the gut wall is leaky, inflammatory mediators can then cross the gut wall into the systemic circulation, causing a systemic inflammatory response, which can then in turn spread to involve the brain, causing neuroinflammation and then neurodegeneration. And what we've shown is that if you reverse this autophagy block using rapamycin, then you can dampen down the innate immune activation in the gut and also systemically and in the brain. And so too, you see a similar effect if you completely eradicate the gut microbiome, thus taking away the stimulus to innate immune activation. And so to conclude, we have shown that loss of GBA in the fly leads to gut dysfunction and widespread innate immune signaling. If you remove the gut microbiome or reverse the autophagy defects in the gut wall, then this dampens down innate immune activation in the gut tissue, systemically and in the brain. And thus we've shown that gut autophagy and the intestinal microbiome are potential therapeutic targets for the treatment of GBA1 associated disorders. And in particular, we've highlighted rapamycin as a potential therapeutic drug for the treatment of these disorders, especially as it is already licensed for use in humans. And so thank you very much for listening. And I would just like to finish by thanking everybody that's contributed towards this work, in particular two highly talented postdocs in my group, Magda Attilano and Alex Hull. And if you have any questions or want to contact me, then please do drop me a line and email me. Thank you very much for listening. Bye-bye now.